Perfect. Welcome everybody to yet another installment of a closer look Open Journal Club in biomechanics. This journal club has been organized for now about a year by myself, Manuel Rausch at the University of Texas at Austin, Emma Lejeune, Matt Bursey, Johannes Weikenmeier, and Adrian Buganza. And it's our great pleasure to invite David Bark today to give us a closer look at a recent paper of his. As a little anecdote, actually, David and I first met when I was an international student at Georgia Tech, and he was a graduate student there. And we didn't interact very much then, but a couple of years later, when I started doing blood clot mechanics, I started reading papers on platelets, and all of a sudden the name popped up again, and we got in touch and met at a conference. And it's just yet another example for how small this academic world is. So for everybody out there who doesn't know this, just be nice to everybody. You never know when you run into whom again in the course of your career. But David has had a really, really interesting academic path, and I would love to hear more about this later. But I think he started with a bachelor's degree at UIUC, then moved to Georgia Tech. And then from there, I believe you first went to New Zealand, then to Australia, and then to Colorado, all for postdocs. And then he took an assistant professor job in Colorado, and now he is actually at Washington University in St. Louis with an appointment at the med school. So I think there's going to be a great conversation opener later to hear a little bit how this path came together and what you've learned from it, etc. But for now, I'd like to invite him to take the mic out of my hand and present on his really interesting paper, as you can see here.
Thanks so much. It's really a pleasure to be here, and I just want to thank you for the invite. It's really an honor. So today we're going to talk about a topic of heart development. And so before we get into that, I just wanted to wish everyone a happy Thanksgiving for anyone that's celebrating. And this is actually a chicken developing from an egg as opposed to a turkey, but we'll just use our imagination. And the reason I thought it'd be relevant to show that is we'll actually be talking about various models of heart development. And one of the major ones is using a chick model. And this has helped us to get a little bit of insight into congenital heart malformations. And so in the image I show on the right, I actually have a chick from another group that they would ligate vessels, and based on ligation, what they saw from these mechanical interactions is they would see these characteristics of congenital heart malformations. So like, for example, ventricular septal defects, and then some other complications. So we'll talk about that a little bit today, and then what we've been doing with zebrafish. So the paper we'll be discussing today is this in vivo pressurization of the zebrafish embryonic heart is a tool to characterize tissue properties during development. And really, the majority of this was done by Alex Gendronellic, who's on the top left there. He's got some amazing technical skills with being able to work with these really tiny zebrafish. So if you look at the image on the bottom left, that's actually a penny, and these are embryonic zebrafish at about 48 hours post-fertilization, so just 2 days after being fertilized. And then on the right is one of Alex's images where he's been able to poke at these zebrafish. And so we're looking at doing a few different things with that now. And then this work was also assisted with by Benesha Zebi, and then Neha and Deborah Garrity. Neha and Deborah are coming more from the biology side and invited us support from over there. And then Benesha helped with some of the finite element analysis, as you'll see. So as mentioned, we just recently moved from Colorado State University to the Washington University School of Medicine. And so this actually happened in the middle of trying to push this paper out, which made things quite interesting, especially because we were working with zebrafish back in Colorado, and then trying to transfer all of that over here was interesting, to say the least. So what is the motivation for the publication? Well, the real reason we wanted to pursue this is honestly because we like biomechanics. So we just had a basic science question on what role mechanics, solid mechanics play in heart development. And so this is what we really wanted to pursue. It also gave us a chance to venture into solid biomechanics. So for those of you who don't know my background, I come more from the biofluids biomechanics side of things. And we work a lot with hemostasis and thrombosis and with medical devices. And so here we're actually venturing quite a bit away from our core areas and venturing over into solid biomechanics. So please forgive me for those solid biomechanics experts out there for what's about to come. And then once we get this study established, we should be able to extend it to many disease states. And so this was really kind of the first effort towards trying to understand the mechanics of heart development. And then we also want to eventually extend this into a model of growth and remodeling to try to predict some of these congenital heart malformations. And if the NIH asks, we're focused on congenital heart disease as opposed to anything else. So we're trying to find solutions to help patients with congenital heart disease. So on that note, we're interested in congenital heart disease because this actually affects 1% of newborn infants. So this is actually infants that have live births, whereas we actually have quite a few that unfortunately pass away while still in the fetal stages. So of this 1%, the leading cause of cardiac arrest remains the highest for these congenital heart diseases very early on. And congenital heart disease remains a concern really throughout development until we get into adulthood and start screwing up our health in other ways with coronary artery disease and some of these other diseases that end up affecting us. But interestingly, with congenital heart disease, only about 15 to 30% of cases has been attributed to genetics. So this means that the majority of these cases have some unknown cause for the malformation or for the disease. And so is a group that's based in mechanics, we're wondering what role mechanics plays in these malformations. And so that's, again, what we're really going to be focused on today. So there's many types of congenital heart diseases. The most common tend to be septal or valvular defects. So on the top here, I'm showing you ventricular septal defect. This is really a hole through the ventricle. On the top right is an atrial defect where we have a hole through the atrial septum. And you have some other diseases as well, some that can be, depending on your perspective, relatively minor, like some valvular diseases. But then some diseases can get highly complex, like Tetralogy of Fallot and Hypoplastic left heart syndrome. And these become exceptionally difficult to treat because the only way to really help the patient is through some complex surgeries. And so this provides us some of the motivation for looking into the mechanics of how these form. And then the surgical interventions for some of these complex diseases are very limited. And when they are successful, which is only a portion of the time, patients often live with lifelong complications and limitations. And the classic example I like to focus on, because this really drives home the importance of mechanics in heart development, is what happens when you have an aortic valve stenosis at the fetal stage. So what we've seen at the fetal stage is these stenoses can eventually progress into something known as hypoplastic left heart syndrome, which is actually where the left side of the heart is relatively underdeveloped. And there's a lot of hypotheses out there on why that might be, including the change in loading on the left ventricle relative to the right ventricle. And I'm going to show you some evidence where that might actually provide some proof that that's the case. But right now, the main way that we have to treat hypoplastic left heart syndrome is through some extremely complex surgical procedures. This starts out with the Norwood procedure, where the right side of the heart is really kind of connected into the left side circulation, because right now the left side isn't really functioning very well. So you call this a univentricular defect. And so after the Norwood procedures, you connect the right ventricle to the aorta. We then go into the Glenn procedure, which is taking the upper body and connecting that essentially in serial with the arterial circulation. And so this helps separate out oxygenated and deoxygenated blood a little bit more. And then finally, we get into the Fontan procedure, which really puts your circulation into a serial arrangement where we begin by pumping blood into the systemic circulation, and then it comes back into the pulmonary circulation. And then after going through the pulmonary circulation, we go and returns to the heart. So these procedures are very difficult, and there's been relatively poor success. But there's been some developments on ways we can potentially improve how we treat congenital heart disease. And one of these methods is to perform a valvuloplasty on an aortic stenosis early on. So what this consists of is inserting a catheter into the fetus and actually expanding a balloon at the aortic valve. And what's been found is you can actually restore the heart function of, or sorry, I should say, restore the left ventricular function where otherwise was relatively playing a very small role when compared to the right ventricle. And with this procedure, it's been seen that roughly 50% of cases end up with a biventricular circulation upon birth, whereas there's still about 50% that continue on with a univentricular circulation where only the right side of the heart's functioning. There's also, of course, procedure difficulties that can lead to death, or just the fact that this disease can be pretty severe, can lead to death. So as a group focused on mechanics, we want to find ways to better understand how procedures like this could help patients and what role mechanics actually play in some of these diseases and potential therapeutics. So as I mentioned, our group has been focused a lot on biofluid mechanics, and we're only recently beginning to look into solid mechanics. So we did a little bit of work looking at hypoplastic left heart syndrome and what the flow looks like in this case. So on the left side, you can see a normal developing right ventricle. And then on the right side is one that's actually experienced hypoplastic left heart syndrome. So it's larger in size, and also the fluid mechanics are changed where the summation of these vortices that are coming in to the right ventricle essentially end up resulting in a loss of energy when compared to a normal heart. So the right ventricle ends up working a lot harder. And there's little to no function of the left ventricle, which is why I'm not showing you what the flow looks like in the left ventricle, because it's not actually relevant in these hypoplastic left heart cases. And then these hearts also experience volume overload and increased wall thickness. We think the increased wall thickness might be related to increased pressure that's required in order for the heart, or I should say for the right ventricle, to pump for the entire body, since right now it's the only ventricle that's really providing blood to the body, and the left is relatively useless at this point. So we're gaining a little bit of insight into defects on the fluid side, and we've also done a little bit with, here we go, with Tetralogy of Fallot, which is a fairly complex disease as well, where you can have a ventricular septal defect, as you see here. And oftentimes you'll have a pulmonary valve that hasn't formed quite completely. So a lot of the flow ends up going through the aorta and up through the systemic circulation. And so in this disease, we also saw some pressure overload. We saw the heart wall thicken. And so this really kind of inspired us to look more into the solid mechanics of heart development and how some of these diseases are forming. Because right now, all these fluid studies are being done after the disease is already formed, and really there's not much you can do at that point. So we want to understand what's going on earlier on. And so to do that, we look at the embryonic side of things. And so normally when our heart develops, it begins just as a heart tube. So that can be shown here for a human at 21 days. Interestingly, for those who aren't familiar with embryonic circulation, the heart tube is incredibly interesting from a fluid mechanics perspective because at this stage, there aren't any valves yet. And this thing is pumping purely through peristaltic-like motion. And it's able to do that without valves up until a much later stage of development at 28 to 50 days where valves begin to form. Also at around 21 days, you begin the looping process, which begins to convert this tube into more of the shape that you think of when you think of the heart. And it begins to lead to distinction between the various chambers of the heart. And then where the valves will end up forming, you actually end up with these endocardial cushions that eventually convert into valves over time. And eventually you'll also begin to join various parts of the heart after this looping process, where you can then begin to see these chambers and valves fully form. And then also in this later stage, you can see trabeculation. So obviously it's exceptionally challenging to look at this in humans because really it's highly difficult to look at the embryonic stages of human development. We pretty much are limited to fetal stages after, I believe it's 14 weeks is usually around the earliest you can really begin to image in the human. And then therefore we have many of these animal models that we can look at. So some common animal models that people have used for looking at embryonic heart development includes the frog, the chick, like I showed in one of the early slides, and then zebrafish, which is really what we're going to be focused on in this work. And mostly all of these species end up developing very similarly through looping, but the zebrafish does have one major difference, which is that they only end up with 2 chambers as opposed to 4 chambers. So there is some limitation with zebrafish in comparison to some of these other models. One other complication with zebrafish is they have some regenerative ability as well. So if you injure their heart, you can actually, the zebrafish has the ability to begin to repair some of these injuries, and that's not seen in some of these other species. And so trying to understand development in zebrafish can be a little bit different. So for zebrafish heart development, again, it goes through very similar stages. So very early on, it begins as a tube, and then it begins to undergo this looping process where it will form a ventricle and an atrium. And then that'll end up being separated by the atrioventricular canal, which you'll end up seeing a valve at a later stage. And then also at the outflow tract, you'll begin to see another valve form. So in these zebrafish hearts, we only have 2 valves that end up forming and 2 chambers as opposed to the 4 chambers and 4 valves that we're used to and know and love in the humans. And then also during this development, we're not just seeing tissue level changes, we're seeing subcellular level changes. So the sarcomere very early on hasn't actually fully formed, and you don't really see it fully form until roughly 42 to 54 hours post-fertilization in the zebrafish. So when we think of the mechanics of the heart developing, we have to keep keep in mind some of these subcellular contributions to how these mechanics are changing in time. So let me turn that sound off. So what people have done with these models is they've begun to look at ways that we can potentially recreate some of these congenital heart malformations. And what has been most successful for this purpose is actually providing mechanical changes to the heart during development. So this really drives home the point that maybe it's not just genetics controlling heart development, but we actually have this constant mechanical feedback on the heart that allows it to know kind of where it's at in development. And that feedback's really critical for normal development. Whereas if you perturb that feedback, you actually can see these malformations. So the classic example is ligation in the chick. So if you basically block the aorta, you'll see ventricular septal defects, much like you would see in a human after progression of certain disease. Also, what's been found is that heart valves, at least in the zebrafish, appear to depend highly on the flow environment. So in zebrafish, before the valves form, you have this low and oscillatory flow that must be there in order for the valves to form. And so if that's removed by adjusting viscosity or changing the heart rate, you actually see perturbed valve formation. So these mechanics are really playing this fundamental role in heart development. Zebrafish have been a little bit more difficult to manipulate during early development. This has to do with the very small size of the zebrafish when compared to other species like the chick. So studies that have actually manipulated mechanics in the zebrafish have been limited to either inserting beads and blocking flow or pushing on tissue during heart development. Or one thing that our group has done is actually expose these zebrafish to a hypergravity environment. So we'll essentially centrifuge the zebrafish, pushing all the blood towards the head. And the idea was that we would alter the preload of the zebrafish. One challenge, though, with this method is we're actually affecting forces throughout the zebrafish and stresses throughout the zebrafish during this procedure. So it's a little bit difficult to say what role cardiac mechanics are having on changes. But what we did see in that model is we saw a change from normal heart development in these zebrafish, where we can see this looping, to this more stringy heart that has this reduced ability to pump flow. So you get this reduced stroke volume. And it appears that these mechanics are really playing a key role at specific stages of development. So if we put these zebrafish into a hypergravity environment very early on, so 24 to 30 hours post-fertilization, we'll see the most drastic changes in development. But if you begin to put them under these altered loads at later stages of development, you actually see relatively normal development and relatively normal function. So there's really this time sensitivity to mechanics, and it appears to really focus around the looping stages of the heart. So with that, it began to inspire us to look at what the mechanics of the heart are during the looping stage and just after looping in these zebrafish hearts. And so, as I mentioned, one thing we've been focused on is biofluid mechanics. So we're able to fully map out the flow and pressure in these hearts. So this is a contour plot of how the pressure varies throughout heart development. And then you can see the velocity vectors as well. And we can extract out the pressure from performing PIV on red blood cells in the heart by basically converting the Navier-Stokes and solving for pressure. And what we saw in these cases is that after we spin the fish, they actually have a reduced pressure and reduced ability to pump blood when you compare it to a normal heart. And so, again, we're able to do this all on the fluid side, but this really gives us a little insight into what's happening with the material properties of the heart. and the development of the heart. So actually, is the heart's pumping very early on, we experience circumferential stresses and longitudinal stresses associated with that pressure development in the heart, even despite the fact that we don't have valves just yet. So to understand some of these stresses and to understand the mechanical properties of that early heart, various groups have begun to probe the heart, no pun intended in this case, because we have an actual probe. So this was labeled the heart poker by the Tabor group, actually here at Washington University. And what they did is they would hold the chick heart in this case, which it's a little bit easier to manipulate when you compare it to the zebrafish heart because it's a little bit larger, giving you a little bit more control. And then they would probe the heart at various locations, and based on probing it, they could extract out some of the properties of the heart. And what they found is this stiffness value, which here it's just the amount of force relative to the amount of displacement of this poker. So it's not necessarily giving us much information on actual constitutive properties in the relationship of stress and strain at this point, but it does give us some insight on where the heart might be stiffer in certain locations. So for example, on the inner curvature where we end up having the heart loop, we can see that the heart is a bit stiffer when we compare that to the outer curvature, which is the outside portion of that heart. And then the sides of the heart have a very similar stiffness as the outer curvature. And then there's actually 3 layers of the heart at this stage. And so there's basically a monolayer of cardiomyocytes on the outside. And then inside that, we have the cardiac jelly, which is just essentially this incompressible fluid you can kind of, or incompressible gel-like material that you can kind of think of. It's mostly composed of hyaluronin. And then on the interior, of course, we have the endocardial cells. And so when this group looked at the cardiac jelly, which is that gelatinous material between the myocardium and the endocardium, they found that that was much more compliant and much less stiff when you compare it to these other layers. And what they ended up doing with this information is they began to look at what's actually driving looping. And we need to know what's driving looping because that's really the first stage of morphological changes that you get as you begin to have chamber formation, septation, trabeculation, valve formation, etc. So the Tabor group was specifically interested in trying to determine what's causing that looping. And what they found is that it really had to do with the differential growth of the cardiomyocytes. So on the outer curvature, the cardiomyocytes are growing much more than the cardiomyocytes on the inner curvature. And they were able to predict this by extracting out the mechanical properties of the heart and by modeling this with some finite element models, both with the simplified model and then they extended it to look at actual geometry of the heart. But one thing that we noticed when we were looking through this literature is that the stiffness of the heart was actually relatively low to what's been reported with cardiomyocytes. So this, the Tabor group found that the stiffness of the heart was about 13 Pascal, and that the cardiac jelly was about a quarter of that. However, if you take cardiomyocytes and culture them, you actually see that they operate much better when you're at the kilopascal level of stiffness. So that's about 1,000 times what they were seeing here. And also with this earlier study, they focused more on single point measurements, and we were interested in trying to expand that and looking at potentially spatially mapping how strain and stress might vary in the heart. So we began to extend some of these earlier concepts to the zebrafish. So apologies for kind of the long introduction, but a lot of that was useful for providing our motivation here. So for the zebrafish, we wanted to apply tensile loads because as the heart pumps, it's usually exposed to these tensile loads as opposed to more of the compressive load like the heart poker on the previous slide. We also wanted to measure properties in the zebrafish because the zebrafish has become one of the most useful models in looking at heart development, especially from the genetics perspective. And there's really not a tool out there to look at zebrafish. And then we wanted a tool as well that would give us the ability to potentially map out spatial variation and constitutive properties of the heart. So what we looked at doing is using an inflation method. So we used an intact heart, and we would take a cannula and poke that into the essentially a region around the atrial inlet, and we could pressurize the heart from that location. One challenge when looking at the heart is normally it's undergoing contraction. So we also applied BDM to stop the heart in order to make these measurements. The tip of the capillary or the cannula was roughly 10 microns. And one problem we had early on was that we'd see a lot of coagulation around the cannula tip. In fact, some of our other work in hemostasis thrombosis, we actually use the same method to create an injury in mice, and we'll look at coagulation around that injury. So this is a very similar thing. So here we ended up applying warfarin to prevent that coagulation from occurring. And then we backfilled our cannula with rhodamine dye so we could actually visualize it and also see when we begin to pressurize the heart. And then we would pressurize it early on using basically a column of fluid. So instead of having some fancy pressure controller, we just used a simple column of fluid, and we were able to change pressure using the equation of rho GH, density times gravity times height. So the higher the fluid, the higher the pressure. There are a lot of challenges with working with the zebrafish at this point in development. So we're working with hearts that are roughly 100 microns in diameter and pretty much in length. And so this becomes very difficult to manipulate. In this particular model, we don't have an ability to separately control axial and circumferential stresses or stretch. So it's very difficult to pinpoint boundary conditions and to fully probe the properties of these hearts. And then, as I mentioned, coagulation is also a challenge with these hearts. So as you might expect, once we begin to pressurize, you could see that the areal changes in the heart began to increase as we increased pressure. We didn't really notice any specific trend, and each of these different symbols represents a different heart. So these are 5 different zebrafish. However, what we did notice is that if we look at the axial relative to circumferential stretch or strain, we found that they vary proportionally to each other, indicating that we might have an isotropic or transverse isotropic property of the heart. And so as we continue on, we're working in cylindrical coordinates where we've got the axial direction going from the atrial inlet to the atrioventricular canal. And then we have, of course, the circumferential direction as well, and then the radial direction going outward. So we looked at 3 different constitutive equations because at this point, we don't really have a well-accepted equation that's been used for the early embryonic heart, and we certainly don't have an equation for the zebrafish. So we assume that the heart was incompressible, homogeneous, isotropic. We assume that cardiomyocytes are really dominating the structural support of the heart and that there's no twisting and no residual stress. So with that, we looked at the Neo-Hookean model, reduced polynomial model, and the exponential model. And all of these assume isotropy and use the first strain invariant. And then for an equilibrium equation, we just essentially started as simple as we could with the Young-Laplace equation, and then extended from there into some finite element analysis. So this Young-Laplace equation was used really for our 2D experiments. So
from the journal club perspective, we had a lot of concerns with this approach. So we aren't accounting for residual stresses, which likely exist during this early heart development, and that's been shown by the Tabor group. We assume local isotropy, despite the fact that sarcomeres probably have some directionality. The heart has multiple layers, and we're really only accounting for one of these layers when we're using this model. And we're also modeling the heart as a continuum. So at this point in development, we have a monolayer of cardiomyocytes, and you can actually see the individual cardiomyocytes kind of lined up. And so we're beginning to approach the limits of what really can count as a continuum. It's really difficult to quantify boundary conditions or the constraints that are on the heart. And due to imaging limitations, we can only look at the atrium, and we neglect three-dimensionality. So using the Neo-Hookean model, we saw some agreement between the experimental data, and we were able to extract out roughly what the material constant would be. So we found that the material constant for the Neo-Hookean model was roughly 32 kilopascal. And we did see quite a bit of variation in stiffness from one zebrafish to another. And we assume that this is actually probably more a limit of our approach as opposed to the fact that there's actual variations in these zebrafish since they're all genetically identical. So this is probably more due to that 2D imaging limitation. We also found some properties for the 2-parameter reduced polynomial model. So here we found the first material constant to be about 49 kilopascal, the second to be about 11. And then for the exponential model, we found similar material constants of 17 kilopascal to 21 within the exponential. And so overall, one thing we noted is that we're working at the tens of kilopascal range versus the earlier work that was done more at the Pascal range. So we're finding a much stiffer result from what was shown with the chick. And then from there, we extended this work to finite element analysis. So we used Abacus, and we ended up, for boundary condition, fixing the outflow tract. And then for the atrial inlet, we made it symmetric about the axial direction. We looked at the max stretch ratio at 10 points along the outer curvature and tried to see how that best matched up between experiments in the finite element analysis. And what we ended up finding is actually that the Neo-Hookean model in this case, in this 3-dimensional case, actually fit the experimental data the best. Whereas in the previous cases, some of these other models appeared to fit a little bit better for 2D. And we were able to do this by taking some confocal images and actually extracting out the full 3-dimensional heart for the FEA model. We also saw maximum principal strains that were roughly between 0.02 to 0.05 along the outer curvature. This was all looked at 2 millimeters of mercury pressurization. Note that in the early heart, we're working with very small pressures, so from 1 to 2 millimeters of mercury, as opposed to like an adult heart, which is more on the order of 120 millimeters of mercury. We did vary the boundary conditions because we had no idea what the actual boundary conditions are for this early heart. So we tried some very symmetric boundary conditions. We tried pinning the atrial inlet. In all cases, we kept the outflow tract of the ventricle fixed, but we were mostly interested in how things varied on the atrial side. But with doing all of that, we were able to roughly find that the stiffness of the heart was on the 10 kilopascal range. We developed an approach where we could begin to extract out the mechanical properties of the zebrafish heart. So again, we had not seen any other studies that really were able to do this. We found that the heart was, again, about 1,000 orders of magnitude higher than previously looked at. And what we did see is an agreement with individual experiments on isolated cells of cardiomyocytes, where groups have looked at aspiration and atomic force microscopy, and they all found that the cardiomyocytes are on the order of 10 kilopascal. We think some of the differences that we're seeing in our work might be due to some other tissue around the heart other than the myocardiocytes. And we also apply an overload condition. So we're working with relatively high pressures for the heart. So we're potentially really stretching out these sarcomeres. And so maybe we're looking actually at material properties of components of the sarcomere like titin. And we're also loading the heart differently than what was done before. So now we're putting it purely under tensile loads. And so that could explain some of the differences that we're seeing. And that led us into thinking about some of the next steps that we're beginning to look into. So we're working on some three-dimensional studies where we can spatially map out the strain and constitutive properties of the heart. We think this will improve the accuracy since it's really difficult to consistently get the zebrafish in the same orientation for these 2D studies. We also have improved ability to control pressure now in the lab. So we've got some better methods than we did initially. And then we also want to look at quantifying residual stress and accounting for that in some of these models. And then we want to extend all of this to a growth and remodeling model that we can apply to better understand some of these congenital heart malformations. One downside with zebrafish is despite any advance we make, we still won't really be able to look at ventricular and septal defects since we only have 2 chambers to work with. So with that, I'd like to acknowledge the BARC lab, which Alex did a lot of this work. We have a few students that have graduated, including one that worked on this project, and a number of collaborators. The one most relevant to this work is Deborah Garrity at Colorado State. And then, of course, the funding agencies that have helped to support this project and the group in general. So with that, I'll leave you with this alien image of the zebrafish embryo. And I'd like to take any questions.
That was absolutely fantastic.
Oh, hold on, I got the volume off. Okay.
Cool. I really loved it. I really also liked the long introduction. I learned a lot of embryonic development that I didn't know about. So I really appreciate that for my own education, and I assume many others. Before we dive into sort of some of the questions I had sent you by email before, let's see whether the audience has any questions. And feel free to ask questions directly related to the study, to the background, or general questions you have for David. Any audience questions?
I have a very quick question.
Sure.
How many cells are in the heart? You had some of the images where you can clearly see sort of the cells. I'm just wondering, in terms of number of cells, how many are we?
Yeah, I couldn't quote the exact number off the top of my head, but if we, for example, just look at this early heart, you can see the individual cardiomyocytes kind of lined up here. So, I mean, I'd roughly guess that's around 20-ish from the atrial inlet to the atrioventricular canal. So again, that's really where like this continuum assumption might begin to break down depending on your perspective.
That's crazy, yeah. Near the end, you had the 3D one that was moving,
yeah.
And then you could also see some of the 3D arrangement. And yeah, it didn't look like that many.
Yeah, no,
yeah, that one, yeah.
Not many at all, no.
I mean, it was just fascinating to see that you don't need that many cells to form a heart.
No, and this is really a fully functioning heart. It just can't produce a lot of pressure. So,
yeah, we're limited to about 2 millimeters of mercury here.
Oh, that's amazing. How many millimeters of water column is that?
A good question. That's probably a good question for Alex. I can't think of the actual value off the top of my head.
I guess quick Google would answer that.
Yes. Yeah, I'm too lazy now and just use Google for my unit conversions.
Oh, absolutely. Is there any other way?
Yeah.
Not that I know of. Those textbooks maybe, but
yeah, it's actually 3 centimeters, 30 millimeters. Kind of cute.
Yeah.
For those of you that maybe are not familiar with measuring pressures in columns, obviously if you have a column of fluid at the bottom, that fluid sort of weighs heavily on that, and that can be measured as a pressure. So the pressure that David is imposing onto these hearts or his students is equivalent to 3 centimeters of water column, which is this much, which you may not even be able to sense with your own hand. Anyways, that's super cool.
No, and actually,
yeah.
that's a great point, and that's one of the things we're most concerned about with this whole experimental approach. Is it's really hard to make sure that we're at 0 millimeters of mercury starting out, and that we're actually, at the pressures we think we are at.
Yeah. Also, I guess you're flowing the water really slowly, but presumably you have a pretty long pipe connected to this, right? And so when you're measuring in such small pressures, the resistance to the pipe flow probably matters.
Yeah, so we wait until the flow actually stabilizes.
So that's one of the beauties of adding the rhodomine into the cannula, is you can see when the flow stops. And so once it equilibrates, as long as we control the height of the fluid column, it actually doesn't matter what sort of path the flow follows anymore, because it only really matters where the height of the free surface is relative to the actual zebrafish. So
makes sense.
As long as we allow it to equilibrate.
Cool. Any other questions by the audience?
I had a quick question. So before I ask, I just want to say hi, David. I'm also at WashU, and I've been meaning to reach out to you and talk to you. So we'll catch up after this. But
that's good.
I wanted to ask about the 3D imaging that you can do, and you were showing the confocal images.
Yes.
So what is sort of the time resolution of those 3D acquisitions? Can you sort of real time measure 3D volume changes?
Yeah, that's a great question. Real time would be a challenge. So there's a potential to use like a spinning disc confocal, which would help us speed this up quite a bit. And we're also looking at potentially using a piezo-based objective where we can actually pinpoint where some of these nuclei centers are at a very high rate. And we're doing this for some other purposes for PIV, but there is the potential to get almost real time, yes. But for this particular study, we definitely did not have that ability. So we were working with resonance scanning, and this was all done on a relatively basic confocal.
Okay. So the image, I kind of asked because the images that you referenced from Larry's work,
yes,
were those OCT based images? They look kind of like an OCT image.
Yes, yeah. So a lot of his work was done with OCT, yes.
Okay.
And then these were SEM.
Okay. Yeah. Okay. Yeah. So we have a benchtop OCT scanner in my lab. So if that would be of interest to you, maybe we could think about something there.
Yeah, that would be cool.
Anyone else? Any questions? One of the things that I find super fascinating about academia, and I try to communicate with my students, is there's many paths to getting where you may end up understanding that you want to be, which can differ from what you think you want to do after your PhD. But there's, I think, a huge bias in a lot of faculty to have taken a very traditional path, right? So most faculty go from PhD to postdoc to a faculty job, which I think is definitely the most efficient way. And so it's nice to see someone who may have not taken an entirely traditional way. And so, David, I was just wondering, what was your reasoning? And I think this can be really helpful to students. And B, and I want to be very clear, right? I mean, it doesn't need to be a professional reason. I love all the places you went to, so presumably that had something to do with it. And then, but also sort of like, what do you see the advantages and disadvantages in taking a somewhat non-traditional way? I mean, you stayed in academia, so it's still traditional in that sense.
Yeah, no, that's a great question. And since I love talking about myself, I'm happy to answer that. So yeah, after finishing my PhD, the normal thing would be to do a postdoc for a few years and either a high-powered lab or one that's very productive, and then go into like a faculty position. But in my case, my PhD was highly focused on computational work and on engineering. And so I didn't have a really good biology foundation or a clinical foundation. So one of the goals I had with a postdoc is really to get that foundation, mostly for the purpose of being able to attract NIH funding, since without that foundation, it becomes much more challenging unless you have the right collaborators. So that was one of the goals with that first postdoc. And another goal also was to really see more of the world. So, one thing that we tend to be limited on, especially in the US, is we kind of see how the US system works and, how academia works here, but we don't really get a lot of insight into how it works in some other countries. So I feel like we're missing out there. So I really wanted to get some experience overseas, and I had this great opportunity to work with one of the leaders in hemostasis thrombosis over in Australia. His name is Sean Jackson. He did some really cool stuff looking at mechanobiology of platelets and how they form a thrombus in some complex flow environments. So that was some of the motivation for there. For New Zealand, that was more of the personal reason of I love the mountains and pretty much everything about New Zealand, including rugby. So that was a very personal choice. And then Australia was more of the professional choice with, personal choice in there, and the fact that it's also a great country to explore. Absolutely loved it there. But one challenge you have is if your long-term goal is to get a faculty position in the US, it becomes very, very difficult if you're overseas. In my experience, I haven't really seen many faculty interviews for overseas candidates unless they're already in the US. So at least when I was finishing up in Australia, one goal was to get back to the US and begin to reform a foundation. The other complication I had is I had been working deep in biology and really kind of lost my roots in the biomechanics world. And so I wanted to reestablish that as well. So that's where I came to Colorado and began to work more with heart valve research and biomechanics, and then could really begin to publish a lot. Was able to get an F-32 to support the work and kind of restart a name for myself within the US. One of the other challenges as well with being overseas is I noticed, like, during faculty interviews, the people I interviewed with didn't really know how to perceive the funding I'm able to get overseas, because it's not their usual funding sources. So is the NIH equivalent to some of these other agencies? It's hard for them to say. So it became a challenge to find the position within the US without being in the US already. It's kind of the long story short, or the more summarized answer. But yeah, that's kind of why I took this kind of roundabout way.
I really appreciate that insight. I did like an industry job after my PhD and then for my postdoc, which I guess, within the context of a pretty homogeneous faculty is already sort of a deviation from normal. One thing that I found frustrating is that I thought that was a really rich experience, and I'm glad I did it. But the about 3 years that I lost kind of come back to haunt you, because a lot of young investigator awards and opportunities are dated from the time of graduation from your PhD. And so in a sense, there's punishing mechanisms that kind of enforces pretty homogeneous faculty demographic. Is that I presume that's something you've experienced and probably also been frustrated with.
Yeah, 100%. So, other than a lot of the kind of typical awards, even just being an early stage investigator with the NIH, your timeline starts immediately when you finish your PhD, or you're doing an MD PhD, whatever your terminal degree is. And in my case, if you think about the funding opportunity, I lost that 3 years when I was overseas, despite the fact that it was an incredibly enriching opportunity. And so I feel like it does kind of discourage exploring, other career paths, or at least other ways to get to your to faculty position. So it was incredibly frustrating, and I'm actually out of my ESI status as of now. So hopefully one of these R01s I've submitted will be get a decent score. But
good luck with that, dude.
Yeah, thanks.
Any other questions by anybody about career choices, career paths, heart development, zebrafish, chicks, turkey? Lots of opportunities for questions.
Yeah, don't think about the heart development when you're eating your turkey.
Or do, depending on how good the turkey is. I guess you can appreciate that it developed into that yummy turkey it ended up being.
Yeah, no malformations.
I have one question.
Yeah.
What since it's so difficult in the academia field to get all these funding sources and such, what kept you in academia versus just jumping into industry in New Zealand or such, or in America?
Yeah, I mean, that's a great question. Really what kept me is probably more of the research questions I continue to have, and kind of the passion for academia and being able to answer those questions. I didn't see the same opportunities necessarily in industry, where you have to focus a little bit more on the money side of things, at least in terms of profit. Of course, in academia, we have money concerns as well. But yeah, so that's part of what kept me in academia. And then kind of what drove me back to the US more had to do with family. And also it's what I knew was, academia in the US. I didn't really, it was kind of a new frontier overseas. And so it was a little bit difficult to navigate their systems. And so, for example, in Australia, they also have kind of a set series of awards and set series of positions that people will take. So a lot of times when they're going into their postdoc, they'll get this award to go overseas for 2 years, and then they'll come back, and then they find a faculty position, and there's specific serial set of awards that you kind of have to get to be successful. So I didn't know how to navigate that, and so that was, again, some of my motivation for coming back.
But yeah, sorry, that was my long-winded answer to that.
All right, no problem. Thank you.
Yeah.
Very cool. I guess we're all getting ready to celebrate this somewhat strange, but by now holiday I've gotten accustomed to of just eating. If there's no other questions, I would conclude this journal club for today. I'd like to thank David one more time. David, I'm glad our paths crossed again, and maybe the next time we talk about thrombus and platelets a little more, which is a shared interest. And then thank you everybody for being here. Thank you for paying attention. And we're going to have another journal club next week, for which Kevin Merman is going to join us. So stay tuned, and we're super excited about that.
Thank you, everyone. And thanks for listening.
Thanks, y'all.